Hello, welcome everyone. Thank you so much for joining us. Fantastic to see many people attending. My name is Katia Pagella. I am Circular Projects Officer at Healthcare Without Harm Europe, and I'll be moderating this session today. I'm going to start with a few bits of housekeeping. So this webinar is being recorded and scheduled to last 1 hour. We have 3 presentations and we have interpretation only for last presentation from French to English. But the questions will be handled in English at the end of all the 3 presentations. So you can type your questions in the Q&A box at any point, and speakers can reply directly or we'll make sure to take them at the end. For those who don't know us, we are Healthcare Without Harm Europe, an NGO working with the healthcare sector to make it more sustainable. And together with our members, we are part of the Global Green and Healthy Hospitals. The network counts today 153 members in 25 countries across Europe, but we also have regional offices and members across the world. So let's get into the session and why this webinar today. We are taking this opportunity and I'm very pleased to announce that we are launching our report, and today we will dive into the main issues that we have framed. So our report is on the PVC-free healthcare to reduce the exposure to harmful plasticizers and to reduce the environmental impact. So the first part is focused on the plasticizer exposure, and then we give an update overview on PVC, and finally a few key actions and substitution initiatives. So many members and healthcare providers have supported us in drafting the report, and I'm sure they are also part of the participants. So today we are taking these few issues and we are going to see them in these 3 presentations. The main topic will be the exposure to plasticizers. As you know, patients often come into contact with these harmful chemicals that can be plasticizers used in PVC medical device that leach out into the body patients, and therefore they undergo to this direct exposure. This presents still a significant risk for vulnerable patients, because they undergo to multiple medical interventions or being exposed chronicle over extended periods, so included infants in the neonatal intensive care unit or the dialysis patients. So we wanted to provide also new data because we are following up the medical device regulation that introduced indeed provisions to help phase out the carcinogenic, mutagenic, and reprotoxic chemicals and endocrine disruptors in certain medical devices. And when it comes to plasticizers, we know that DHP, the most conventional phthalate, for which now we need the specification to justify the use above certain limit, but still not completely banned, we can still find it in the market. Manufacturers are anyway taking steps to replace these DHP plasticizers, but we want to question if indeed are we going in the right direction, if these are indeed true safer alternatives, and what are, these alternative plasticizers. So we'll see in the second part of the webinar 2 presentations from the academia to better explore the exposure from DHP and the plasticizers. But on the other side, before jumping to this exposure to plasticizers, in the first presentation, we will also take a look to the polymer itself, so to the PVC that is widely used for medical devices, and actually it also represents some other impacts on the environment and human health through all the life cycle. ECHA is currently working on the investigation report on PVC and its additives, so we also thought that this is the case to provide new data and new evidence on the impacts of PVC and plasticizers. Also, we are organizing our conference on sustainable healthcare, CleanMed. It's taking place from 5th to the 9th June. And so you will find a lot of sessions on climate, safer pharma, and circular healthcare. We'll also have roundtables and workshops. But if you want to learn more about the PVC-free healthcare alternatives, we are also organizing a workshop on the 5th June and 7th June. It's the same workshop, but we are replicating in 2 days. So this is the occasion also to learn more about the alternatives, to understand the market, and to really try to have an interactive session and interact with the suppliers. So I'm pleased to introduce the speakers. Ted Shetler, he's a MD and PH and scientific advisor to Healthcare Without Harm US and Canada. He published many articles and served as advisory committee to the USA APA and National Academy of Science. Ted has also given a huge contribution in helping us in drafting the report. And today he will give, as I mentioned before, the big picture on PVC to better understand what is this polymer, what are the impacts, and why also it requires so many plasticizers and additives. Then we will hear from Lucas Panel and Pauline Claes from the University of Antwerp. They will share their studies on the neonatal exposure to phthalates and alternative plasticizers via parental nutrition. Lucas is a medical doctor and pediatric. He's pursuing a PhD on the exposure to plasticizers in the neonatal intensive care unit with a grant from the Research Foundation Flanders. And Pauline, also PhD, she focused her work on the analytical, environmental, toxicological aspects regarding the human exposure to phthalates and alternative plasticizers. And then in the end, we will have a presentation from Lise Bernard. She's a pharmacist at Clermont-Ferrand University Hospital in France and professor assistant. Her studies focus on the medical device to the risk associated, and in particular, those by leaching out of plasticizers from PVC. And today she will present her findings and current studies on the plasticizers, and again, DHP and alternative plasticizers. So without further ado, I'll turn it over to you, Ted. Thanks.
Thank you very much, Katja. It's a great pleasure to join you today. As Katja mentioned, my task is to give a brief summary of the environmental and health hazards of polyvinyl chloride across the life cycle. I'll start by mentioning that the PVC does have some advantageous features, including its cost, versatility, in that it can be made either flexible or rigid, depending on the application that's being designed. And it has strength, and that is one of its attributes. On the other hand, there are public health and environmental hazards of PVC throughout the life cycle, beginning with production, use, and disposal, including some of those that I've listed here on this slide. Leaching of plasticizers has been mentioned. We'll talk more about that later. Leaching of hazards from landfills when it's disposed of, from incineration when it's disposed of, and then there are some recycling problems that we'll briefly discuss. And I also want to clearly mention that it complicates the recycling of other plastic polymers. Polyvinyl chloride is produced with a number of fillers, stabilizers, pigments, plasticizers, lubricants, flame retardants that vary with the application and the functional needs. And some of these are used in fairly high volume in the polymer. The stabilizers initially, when PVC was in large use years ago, were lead and cadmium. Those have largely been phased out. They're prohibited in some countries. They've been replaced with others, such as calcium, zinc, and organotins. And the plasticizers are variable, and we'll hear more about those from the other speakers. This is just a diagram of the production process for PVC, beginning with either natural gas or petroleum hydrocarbons that are used to produce ethylene. And the chlorine is derived from the electrolysis of sodium chloride brine. The two of them are combined to form ethylene dichloride, which is then converted to a vinyl chloride monomer, and then into the polymer polyvinyl chloride, at which point various plasticizers, stabilizers, pigments, and fillers are added depending on the application that's intended. Now, here in the bottom left of this slide, I've shown you the various processes that can be used to generate the chlorine from the brine. Mercury processes have largely been phased out. What we're using mostly in the US and in Europe today are diaphragm processes. Although even in Europe, the asbestos-containing processes have been largely phased out, whereas they're still being used in the United States. And then there are membrane processes, which I'll describe in a moment. I'll briefly mention that there's another process for producing PVC that's largely used in China, which is acetylene-derived, in which the acetylene, which is derived from coal, is then reacted over a mercuric chloride catalyst to yield vinyl chloride. This results in a fair amount of environmental contamination with mercury. And as I mentioned, this is largely confined to China, not being used, as far as I know, in the US or in Europe. To make chlorine from brine, you need to use either an asbestos-coated diaphragm or a membrane that's coated with perfluorinated chemicals. I've listed some of them here in this bullet in the middle of the slide. The mercury cell technology has largely been phased out. This is a graphic depiction of that process, which is fairly energy intensive. The chlorine comes in, the brine comes in to the anode side of the fuels of the electrolysis cell. Chlorine is generated. The sodium then goes across the membrane or the diaphragm into the cathode side of the cell, where it is converted into either caustic soda, sodium hydroxide, or to hydrogen. So that's the general process that's used for generating chlorine from brine. There are hazardous wastes and byproducts that come from this process. There's brine mud that comes from the brine purification. And then the diaphragm and membrane cell processes generate their own solid waste, including the diaphragms and the membranes, which after they're used for a while have to be disposed of, and they're largely discarded into either landfills or the typical place for disposing of them, where they can represent ongoing risk. For example, asbestos is a carcinogen, and perfluorinated compounds are highly persistent and have a range of toxic properties depending on the actual molecule. So these are then in the environment where they can leach and cause additional problems. Now, I've mentioned that ethylene dichloride is generated from the ethylene plus the chlorine, and then that's converted into vinyl chloride monomer and polymerized. So the ethylene dichloride, which is an intermediate here, is highly flammable. It has, with acute, fairly high level of exposures, impacts on the central nervous system, liver and kidneys, causes respiratory distress and cardiac arrhythmias. And with chronic exposures, it increases cancer risk. It's classified as a probable human carcinogen. Vinyl chloride monomer, which is derived from the ethylene dichloride, is also highly flammable. With acute exposures, can cause damage to the central nervous system, liver and lung. And with chronic exposures, liver and kidney problems, and it too is it's a known human carcinogen able to cause a variety of human cancers as listed here. These observations are derived primarily from occupational exposures historically, and as this information became available, efforts were made in the workplace to reduce workplace exposures to these chemicals during the production process. And they have been successful in many settings. However, the general public nearby the facilities that are making these chemicals and from their intermediates are also at some risk, because there's a fair amount of release of fugitive emissions from these facilities. And I've given some figures here for the amount that's released from US facilities. where they report releasing about 400,000 pounds of ethylene dichloride and vinyl chloride into the air each year. About 20 years ago, there was about twice that amount that was being emitted from European Union facilities, and I have not been able to find more current figures on fugitive releases. The other thing to keep in mind is that these chemicals can be accidentally released with accidents during transport, as you may have heard about with a train derailment in eastern Ohio earlier this year, where 5 cars in a long line of cars were derailed, and ultimately the vinyl chloride in those 5 cars that were derailed and damaged had to be burned, releasing the vinyl chloride and its combustion products into the community environment. Now, there are also byproducts of ethylene dichloride production that contain a number of chlorinated toxic compounds. And in the United States, data show that over 4 million pounds of hazardous waste coming from ethylene dichloride manufacture was shipped as for waste disposal in other locations. Most of this waste is burned in incinerators and in cement kilns in the United States. So it's a fairly large amount of hazardous waste that's produced by ethylene dichloride production. Then finally, I talk a little bit about disposing of polyvinyl chloride. Incineration and landfill are 2 ways that have historically and continue to be used, although landfilling of PVC waste is not allowed in some countries. It should be noted that additives can leach from the PVC and contaminate the surrounding environment of the landfill. With incineration of PVC waste that produces toxic gases, including hydrogen chloride, and also we need to keep in mind that highly toxic dioxin and furan production can happen depending on the incinerator design and operating conditions. Pollution control equipment will capture some of that, obviously, and it may be then in the ash of the incinerator, which must be deposited in the ash, which must be disposed of. Recycling of PVC is an important topic you can consider. First of all, PVC must be separated from mixed plastic waste because it complicates and impairs the recycling of other polymers. If more than 8 or 10% PVC in a mixed plastic waste stream can destroy the recyclability of all the other polymers that might be in that waste stream. Well, once PVC has been separated, it can be ground and shred and with the intent of turning it into a recycled product. But this often results in downcycling because the quality of the PVC is not sufficient to recycle at a high level. This is mostly true for products other than windows and pipes, which have been successfully recycled back into windows and pipes. Then there's chemical or feedstock recycling to consider. This is an attempt to try to melt the PVC at a fairly high temperature, often in a low oxygen environment, to recover chemical components from it. But this has been largely unsuccessful economically and has not been scaled up at all. I'll just give you some figures here showing that in 2022, about 813,000 tons of PVC waste were recycled in the European Union. In the European Union, about 2/3 of that was pre-consumer waste, and only about 1/3 was post-consumer waste. And this collectively represents about 12% of the total PVC produced in Europe. Now, there are alternative polymers. We'll hear more about some of the alternatives later. They depend on the application and the needed functional properties. I've listed a few here. The polyolefins, such as polyethylenes and polypropylenes, are adequate and sufficient for certain applications. Others to consider are ethylene vinyl acetate and silicone. They'll vary again with the functional needs that are necessary. These generally avoid many of the high volume problematic additives, including the plasticizers. And I'll finally close with just a comment about the concept of a plastic hierarchy. Here, what I've done is summarize a plastic hierarchy coming from the Scotland National Health Service. Plastic hierarchy is a way of prioritizing polymers that are more suitable and prioritizing other polymers for reduction and phase out. And the criteria that can be used to help generate this kind of thing is thinking about a combination of hazard assessment, circularity, and necessity, where we're seeking to retain value in plastics by ensuring they remain active in the economy with high quality recycling and encouraging recycled content, giving preference to plastics that can be recycled to a high standard, and reducing and phasing out those that cause the most significant damage to the environment and human health, and reducing unnecessary plastic use where possible, and where single use is unavoidable, making sure that they maintain a high minimum of recycled content and are themselves fully recyclable, and decreasing packaging as much as possible. These will help us decide among plastic polymers as we're making purchasing use decisions, trying to move to a safer material economy. And I'll stop there, Katya. Thank you very much for the opportunity to be here today.
Thank you very much, Ted. I know that we would need more time to talk about PVC, but that is very comprehensive overview. Indeed, and now we'll understand the exposure part on PVC. So let's look at the plasticizers, but it was really a good overview to understand the impacts on the PVC itself, on the production and disposal. So Lucas and Pauline, if you're, yeah, okay, I'll give the floor to you. Yeah, thanks.
Okay. Let's see. Normally you should be able to see my screen.
Yeah,
like
perfect.
This.
Perfect.
Okay, so first, thanks Healthcare Without Harm for having us, and of course audience to be here. Thanks Katja for the introduction, and Ted for the more general introduction on PVC. We are Lucas and Pauline, and on behalf of the Greater Plastic and ICU Research Group from the University of Antwerp and the Antwerp University Hospital in Belgium, we will focus a little bit more on exposure in practical care in the neonatal intensive care unit, and we will present and discuss our recent findings on plasticizer exposure, mainly in parenteral nutrition in the neonatal intensive care unit, as we recently published in the International Journal of Pharmaceutics. So to start with, two main concepts of our presentations, which are already a little bit explained by Katja and Ted. Plasticizers are a heterogeneous group of chemical compounds added to rigid plastics as PVC to increase flexibility, durability, and so on. And are used in, apart from many plastic products, also in a lot of plastic medical devices, as you can see here. Nevertheless, due to their lipophilic nature and non-covalent binding to the matrix of PVC, DHP and other phthalates have been reported to leach from plastic products into the human body, as shown by several biomonitoring studies in adult intensive care unit, pediatric, and also neonatal intensive care units. And another problem, apart from them leaching, is, as mentioned, the hazard profiles of phthalates, which can vary, but mostly include hormone disruption and reproductive and developmental impacts. And therefore, most of those phthalates have been, some phthalates are included in the candidate list of substances of high concern and have been listed as endocrine disrupting chemicals or carcinogenic or mutagenic or toxic for reproduction. And in 2017, the EU adopted the medical devices regulation in order to phase out phthalates such as DHP from medical devices, and declaring that their use above a concentration of 0.1 weight % was subject to justification and or labeling. And this knowledge on their toxic aspects and the banning from medical devices sped up the search for alternative plasticizers. So that on the concept on plasticizers, and on the other hand, a lot of care, but especially care for newborns, premature newborns. In a neonatal intensive care unit, this care relies on several indwelling plastic medical devices with a fundamental role in respiratory support, intravenous catheterization, nutrition, and an example is also parenteral nutrition, which is nutrition given by a catheter, either peripheral or central. And this nutrition is stored in plastic bags, is administered through plastic lines, through a plastic catheter in the patient. And previous studies have shown that it is potentially a major contributor to high phthalate and or alternative plasticizer exposure in the neonatal intensive care units. And this is especially important that in the first weeks of care for premature neonates, all or most of the nutrition is through parental nutrition. So premature neonates are potentially exposed to high levels of those phthalates. Therefore, the primary aim of this study was to investigate to which extent parental nutrition contributes to current phthalate. So the study was executed in 2020 to 2022 to current phthalate and alternative plasticizer exposure. And the secondary aim was to estimate a potential health risk of this exposure. And now give the floor to Pauline to talk us through the methods and raw results.
Yes, thank you. I hope you can all hear me. I will go over the different parts which this project was split into. So we mainly divided the setup in two parts. The first one where we quantified which phthalates or alternative plasticizers were present in the plastic medical devices which are currently used for the parenteral nutrition administration in the NICU. And then the second one where we studied the amount of phthalates and alternative plasticizers which leach from those plastic medical devices during the clinical administration in the NICU. So these ex vivo experiments simulate in vivo parenteral nutrition administration, and we make an estimation for a neonate of 1 kilogram at postnatal day 0 to 5, which was administered 120 milliliters per kilogram per day. This parenteral nutrition was prepared and delivered by the Antwerp University Hospital Pharmacy, as it also is in the clinical real-life situation, consisting of both non-lipid solutions and lipid emulsions. And samples were taken before the infusion and after time point 12 and 24 hours. Then we go to the results of phase 1. On the next slide, yes, thank you. And here you can see it's a big table, all the concentrations of the plasticizers in the medical devices themselves. We see that DHP and several alternative plasticizers are present in relevant amounts in the different plastic medical devices. A total of 14 samples were taken from 5 plastic medical devices, and they are quantified for 17 phthalates and alternative plasticizers. However, we see a broad range of concentrations from 10 nanograms per grams to several milligrams per grams, which is why we expressed the concentrations in mass percentages. Compounds with a mass percentage above 0.1% are seen as real plasticizers, and substances below 0.1% as trace compounds or impurities. So we see in this table a predominant use of ATBC and TOTM in the plastic medical devices, which are manufactured in the last 2 years. And this is in contrast with results from a study in 2018, where we see a predominant use of DHP. And this was in plastic medical devices used in Belgian and Dutch pediatric intensive care units. We see a clear variation in concentrations, but also in combination of plasticizers, which are detected in these different parts of the medical devices. In the more flexible parts, we see a mixture of compounds that are detected, while for other medical devices parts, only trace compounds were identified. So I think it's remarkable that both the pressure sensor discs were mainly plasticized with ATBC, which is an alternative plasticizer, and to a lesser extent with DHT and DINCH, in contrast to the inline filters, which seem to contain no plasticizers except for low traces of ATBC. In addition, the extension of the extension line used for the crystalloid infusion contained a higher plasticizer concentration of DOTM compared to the lipid administration, which mainly contained ATBC. I think it's also remarkable that, or we should mention that these medical devices were all labeled to be DHP free, while no information is found on the presence or absence of other plasticizers. So there's only information for one phthalate, which is DHP. It is still detected in 4 of the analyzed parts, but only in concentrations below 0.1%, except for one medical device part. It is also noticeable that both DHP and DHT were found in the plastic medical devices which are plasticized with DOTM, and this can be the consequence of impurities of a mixture of DOTM which is used. Then on the next slide, again, some tables about the results of the second part of this study, which was the ex vivo leaching experiments, which demonstrated that the lipid emulsions contain significant amounts of both DHP and several alternative plasticizers, such as ATBC, DOTM, DHT, and DHA, while hardly any plasticizers were detected in non-lipid solutions. So to go over the table, our results, our results show that we see trace amounts in plastic medical devices of DHP, but DHP itself was detected in considerable concentrations in all lipid samples, while having very low concentrations in the crystalloid sample, so in the non-lipid matrix. So we see that there's also no consistent increase of DHP after the first sample, which is taken before the start of the infusion, which leads us to assume that there's actually little or no DHP which actually leached from these plastic medical devices themselves, but that it's maybe a source within the lipid emulsion itself. Then 3 other phthalates, DMP, DEP, and BBZP, were detected in most lipid samples with a high detection frequency, with concentrations similar at all time points, while it's absent or at very low concentrations in the non-lipid samples. In these non-lipid samples, we see DINP and to a smaller extent DIDP, but also with low detection frequencies in the lipid samples. So similarly to the DHP, we assume that the source of DINP in the non-lipid samples is maybe this infusion solution itself, as we don't see an increase of the concentrations in function of time. And then the leachability patterns or the leachability profiles of the different plasticizers next to each other. So these concentrations show the concentrations in the lipid samples in the beginning of the infusion and at time point 12 hours and 24 hours. And here we focus on the lipid samples since the detection frequencies in the non-lipid samples was quite low, so there we don't see any clear leachability patterns. So I will go from left to right, starting with ATBC, which is an alternative plasticizer, as we saw 2 slides ago, used in various medical tubing, showing significantly higher migration potential compared to the other alternative plasticizers. And this could be due to its lower molecular weight or a smaller steric conformation compared to the other compounds shown in the slide. Our results show that ATBC was present in high concentrations in the pressure sensor discs used in both the non-lipid and the lipid circuits. And it was also the main plasticizer in the extension set, which was used for lipid administration. So although being present in the lipid emulsion at the starting point, we see a clear increase in function of time, indicating that this compound leaches from the plastic circuit into the lipid emulsion. Then the second alternative plasticizer is DHA, which is reported to have a very high migration potential, the highest of all alternative plasticizers. But we see this alternative plasticizer only in very low concentrations, and we don't see any real evidence of leaching in function of time. Then we go to DHP, which is the reference point, and we compare the different plasticizers to this molecule. So DHT is a variation of DHP, which is chemically a little bit different, but very similar. And it showed that the alternative plasticizer DHT can leach 18 times less than DHP into the lipid emulsions. And this is also confirmed in our results that DHT concentrations actually decrease over time. So this leads us to assume that there is maybe resorption in the polymer matrix of the extension set, or maybe adsorption or absorption to the plastic materials, which have also been described previously by different types for different types of drugs. Then TOTM, which is also a very interesting alternative plasticizer, which is increasingly used the last years in blood transfusion and infusion sets, based on its lower migration potential due to its high molecular weight and also because of its hydrophobic characteristics. Several studies confirmed that TOTM the migration potential is lower compared to other plasticizers. For example, comparing with DHP, it's 200 times less likely to leach, and DHA 100 times less likely to leach, making it a more suitable choice as plasticizer. And this is also confirmed in our study, where we see virtually no concentrations of TOTM in the non-lipid samples, despite it being a plasticizer which was highly abundant in the plastic medical devices which were used for the administration. And although the concentrations were quite low in the plastic medical devices used for lipid administration, we see very low concentrations during the migration study, so very low concentrations in the lipid samples during administration of the parenteral nutrition. So I think this was the main characteristics of this slide. So I will now give the word back to Lucas for the clinical.
Okay. And before finishing, thank you, Pauline, for discussing the results. And we also want to know what is the potential risk of this exposure. And for now, for risk assessment, what we can do is estimate daily intake. So calculate based on the total volume that the children would receive in practice over 24 hours. And hypothesize for a child to be 1 kg, as we said in the methods. And then we can compare the estimated daily intakes on the right column with the reference doses or the tolerable daily intakes or derived no effects levels. And then we can see that for all phthalate and alternative plasticizers we detected in our studies, the estimated daily intakes are below the reference doses or the derived no effects levels. Even for ATBC, for which we have estimated daily intakes of almost 100 microgram per kilogram per day. This is lower than the current, I'll say that, current reference doses that are there. So if you see this slide and comparing to previous studies showing estimated daily intakes of up to 1,000 or 10,000 microgram per kilogram per day for DHP, we can see a favorable evolution. Nevertheless, even though single compound estimated daily intakes are below the reference doses, there are some remarks to take into account before making full conclusions on safety of neonatal exposure. And first of all, as shown in our study, results show simultaneous exposure to different hazardous compounds at the same time, potentially causing an additive effect as suggested for different phthalates. And one approach is to sum estimate the intakes and compare them to one reference dose. And so you can calculate an hazard index, which was still below 1. In this study, this was about 0.35. So which suggests that the current exposure is below the reference levels. But secondly, those reference values are those toxicological endpoints of all those plasticizers may result from animal studies that are afterwards extrapolated to a healthy adult human population after oral exposure, which is rather different than intensive care exposure, especially in premature neonates. And first of all, exposure is 100% intravenous, which might lead to higher bioavailability. Secondly, those premature neonates are in a critical developmental period, both for brain maturation, lung maturation. And secondly, they have an immature metabolism and excretion. And for example, human phthalate metabolites are mostly glucuronide conjugates, and which are therefore not biologically active. However, glucuronidation is a process that happens in the kidneys, and it can be significantly lower in premature neonates, which often have lower glucuronil transferase activities, and which might lead to increased toxicity. And in addition, the toxicological endpoints of those studies are mainly on reproductive and endocrine disrupting toxicity. But more and more epidemiological studies show associations with respiratory health, neurodevelopmental outcomes, and for those there is insufficient human data on the effects, and especially for premature neonates. And yeah, lastly, as I slightly mentioned, for ATPC, even though the exposure is up to 10 times higher than DHP or some phthalates, it's still below its safety profile, but its safety profile is based on way fewer studies than other plasticizers. So we should be sure, in cooperation with manufacturers and legislative bodies and end users as us, that we don't change for regrettable substitutions. A last slide before ending our presentation is quickly on the legislative gaps, both on the local and national level. In Belgium, from our hospital, neither from the government, national government, there is no information on the use of plasticizers or other carcinogenic compounds used in plastic medical devices. They refer to it should be safe to contain foods, and it should be in line with the EU medical devices regulation. EU medical devices regulation, as said, only describes or limits the presence of 4 phthalates and gives no information on alternative plasticizers, on safety and labeling. And maybe we can hold that for discussion. And there's also the problem of the transition period, currently until May 2024, which might be postponed even until 2028. And it is interesting to note that regarding the use of these medical devices specifically for nutrition, most plasticizers are like ATPC, DHT, and DHA are authorized for use in food contact products, while there is no information either in the medical device regulation nor in the guidelines from the EFSA on the use of TOTM. Lastly, awareness among clinicians, as a survey in 2019 showed that end users like us, like nurses, doctors, and so on, have even less knowledge on the presence of phthalates and even less on the means to diminish the use of harmful phthalates in the NICU. To conclude, our data indicate that premature neonates requiring parental nutrition are still intravenously exposed to DHP, albeit lower than in previous studies, and to a range of alternative plasticizers, and making a target for reduction, as shown in the tables by Pauline. And ATPC seems to leach significantly from the devices, while TOTM might be present, but as leaching very, very low amounts. Probably being a good alternative plasticizer. Exposures mainly in lipid compounds and less in crystalloid compounds. Current hazard estimation shows it could be safe, taking into account that the toxical endpoints might not be suitable for premature neonates, and that we should discuss the ways to fill in the legislative gaps. Okay, thank you for your attention. I hope everything was clear. I'm sorry for
Thank you guys.
No,
thank you guys. It was very clear. Let's go to Liz, and then we can come back maybe again on your conclusion to see also the findings of Liz. Thanks, guys. Liz, the floor is yours. Just a reminder for the participants that if they want also to listen the French version, they can click on the button on the interpretation in French. Otherwise, they can stay here in the English version. Thanks.
Right, good afternoon, everyone, or good morning. I would like to thank Healthcare. I'd like to thank Healthcare Without Harm, particularly Katja and Romain, for inviting me in this webinar, which deals with a very interesting question, which is to reduce toxic plasticizers in medical devices. Thank you, Maria, the interpreter. I do speak English, but I wanted to have an interpreter so that you may better understand my presentation. So today I'm going to talk to you about the work which we carried out on this topic, but beforehand I would like to present our research activities. So our research, it is a transnational type of research focused on medical devices, aiming to reduce interaction between the medical devices and nutrition and the environment itself. And this interaction really is part of two very distinct phenomena. First of all, the leaching of compounds which are in medical devices, additives, what we call additives, additives release. And when we talk about medical devices, of course, in terms of PVC, we talk about the fact that a lot of these additives are plasticizers, and they leach and they lead to toxic risks, toxic hazards for patients. Toxic risks which are added to already established risks that we have for some patients. And in fact, some of them are endocrine disruptors, such as DEH. Now, the second phenomenon is the drug absorption, the general absorption, of drugs in medical devices or via medical devices, which could lead to an inefficient therapy. And this risk of interaction between the container and the contents is higher with PVC medical devices. You can see it in quite a lot of literature that on the subject, which has been published for quite a few years. And the important challenge is that these two phenomena in clinical practice cannot be separated, or not separated, so to speak. So looking for solutions should enable us to really simultaneously answer these two issues. So how do we study these phenomena? How do we find solution for the safe care of patients? Well, we have various successive approaches or simultaneous approaches, but there is a fundamental approach, and we have ex vivo models which enable us to take stock of some situations which cause issues, release of additives, identification of the parameters, assessment of phenomena, drug losses, influencing parameters, material analysis, and an analysis, a proper analysis of the material, which is very important, and the dosages to which patients are exposed. And we also developed a molecular simulation with other teams within our research institute, which enable us to have an additional approach in order to study this phenomenon properly. And the other approach is a clinical approach, which is with clinical studies and with ex vivo studies, ex vivo models, and we also have in vivo studies of bio monitoring, which enable us to assess real life risks associated to toxicology assessments. So these, well, very quickly, this is the extent of our research. And in terms of drug absorption, we work on models which use various known active principles with various physicochemical characteristics such as diazepam, paracetamol, insulin, which are used in an environment which reflects really very frequent use of plastic. And the models enable us to study the various dilution solvents in the excipients and to really have a look at the absorption phenomenon of all the components of the compounds of the formulation of medicines, particularly excipients. Now, the absorption phenomenon is very important, and we saw it with PVC medical devices, but it is also possible to have a release with other materials in syringes, polyethylene, which we find in longer tubing, for example, in taps, or polyurethane and silicone, which you can find in catheters. So we also assess the interaction between container and contents with packaged articles, for example, for the preparation, the hospital preparations files that we have. So tubings, and PVC, and we look at PVC alternatives. Let me give you an example. Insulin. Insulin does lead to interaction with lots of materials, and of course, there is interaction with PVC, but also with polyurethane, and the risks are high, particularly with a compound like insulin, because insulin is a very narrow therapeutic medicine, especially when you are talking about vulnerable populations such as neonates and intensive care babies. So here are examples of ex vivo models with this absorption model. Following our research, on the right-hand side, you have the developments of diazepam concentration and insulin on the right during their passage through catheters from various pharmaceutical laboratories. The first three polyurethane and the last one is silicone. And you can see that you have very different behavior relating to the makeup of these medical devices. And on the left, you also see the development of insulin concentration in dynamic conditions, infusion, perfusion, through tube extensions and via an electric pulsar. So you have 10 minutes per hour on the right, and insulin was in a perfusion type with various extensions of different makeup with different materials, PVC for one, polyethylene, polyurethane, and others, and TPO, thermoplastic olefin as well. And in the same way, you can see that depending on the constitution of the compound, the interaction will present different results and a more or less important degree of absorption relating to the flow and the medical device itself. Now, the second heading, the second part of our research deals with the migration of additives from medical devices. We really worked a lot, of course, we worked a lot on leaching out from medical devices, and we saw that all plasticizers don't release in the same way from PVC tubing, although the tubing was actually placed in the same condition, and the migratory behavior is different in proportions and in and over time. And you can see that DEHP presents the worst profile with growing evolution, increase of the rates which are leached over time in proportion with and also with higher releases than other plastics and plasticizers. We built models as well, which enabled us to reproduce clinical environments, some clinical environments in a hospital setting. And we were able to assess the dosages the patients are exposed to. So you have very different models. You have an ECMO model, you have an hemofiltration model, you have an infusion model for an adult and for the infant, and a respiratory assistance model, which you can find. You can find all this information because it has been published. So the clinical approach is relating to this content container interaction was developed, published through MSM, the National Medicine Safety Agency, ARMED project, and the ARMED NEO project between 2015 and 2018. Now, for the ARMED NEO, we actually concentrated on high-risk neonatal population. As Lucas said, the neonate population is very vulnerable, highly vulnerable, because their physiopathological context is such that it is a population that's included within the exposure window, because they're much more sensitive to particularly endocrine disruptors, particularly when they are in the intensive care unit just after their birth. And of course, mini puberty period is also affected by testosterone levels, you know, chemical pollutants such as plasticizers can affect testosterone levels and can lead to all sorts of diseases, impairments, and behavioral impairments and genital impairment. And they are also overexposed due to the nature of the medicine that they take, the concentration of the medicines, the very low flows which are set in order for medicines to be administered, the temperature in the incubators, and of course, let's not forget that they are subject to, they are, you know, tied to all sorts of medical equipments, and of course they can't really excrete or detoxify very easily. Now, the last part is, you know, of our research is the toxicological approach within the framework of this medical devices and content container interaction. And in ARMED and ARMED NEO, we actually have some toxicity rates through tests that we carried out. You know, we did cytotoxicity studies, and there is a difference of toxicity between plasticizers and a more favorable endocrine disruptor levels but it's true that those tests have been carried out on molecules only, and we look at what we know about molecules, compounds such as endocrine disruptors. We know that a combination they have very cumulated effects. There are cocktail effects indeed from these plasticizers. Sorry. Now let me talk about the Ahmed NEO study, which is a clinical trial, which is observational, prospective, and multi-center, concentrated on two investigating centers in France, the Clermont-Ferrand and another university. At another university, you could see that patients which were enrolled in the studies were supposed to be in hospital for at least 48 hours. These patients were under catheter, urinary catheter, and they were subject to at least one risky situation: parenteral nutrition, hemodialysis, ECMO, and patients who could also have perfusions. The exclusion criteria were as follows. The criteria, the exclusion criteria included the patient death or removal of the urinary catheter, or if we had anuria during the study. There's the design of the study, which was carried out. Patients were in intensive care units, they were included, and every day, and we'll look at the detail later on, we would collect all the clinical information concerning the medical devices which were used for their care, and together with urine samples, which were actually carried out every day at the same time, and which were compared to the exposure on the previous day. And in fact, patients could have a participation time of 14 days maximum, and at the end of the study they became their own witnesses with a last urinary sample in the urology ward. In fact, so they were away from the intensive care unit, and this design enabled us to compare populations which were rather homogeneous, one exposed to plasticizers in medical devices during in an intensive care unit, and the other just coming out of hospital in neonatology. As I said before, every day there was a measure of the rate of the metabolites in the urine of children. So in the intensive care unit every day for 14 days, and then in neonatal care unit before discharge. Now, before we had in the project Ahmed, we had developed the dosage methodology through chromatography, which is an MS detection. And that enabled us to carry out assessment of plasticizers and metabolites, particularly DEHP, DEHT, and TOTM. We also did an inventory and recorded all PVC medical devices used for the care of patients when they stayed in hospital, the number, the type, the model. We also recorded the demographic data of these patients and anthropometric data, age, gender, body weight, and the type of incubator, closed incubators, etc. So these are the results which we obtained out of a total of 97 patients over 16 months. You can see that it's a population rather homogeneous as to their demographic characteristics. So a gestation age of 36.1 and a weight of 2.5. But when we had clinical settings for patients on day one, you have a few differences. The majority, practically all patients, were benefiting from parenteral nutrition, 37% for Center 1, parenteral nutrition, and you can see the percentages for Center 2. Also, you have respiratory assistance, 100% for Center 1, 84% for Center 2, and 15 blood transfusions for Center 1 and 5 for Center 2. 508 urinary samples were collected, and the last sample was collected for 39 patients in Center 1 and 12 patients for Center 2. Now, the assessments of plasticizers metabolites in urine showed that the exposure of newborns to DHP was 5 to 10 times higher than that of the DEHTP and 50 times higher than that of the TEHTM. For each plasticizers, there was a predominance of the specific biomarkers of exposure, the 5CX MEHP, 5CX MECPT for the EHT, and the 5CX MEHTM. And we also showed that the urinary metabolites of DHP and DTM were significantly lower when patients got out of hospital. So the last sample comparing to the rate measured during the time spent in intensive care unit with a factor of 18 to 35-fold for DHP and 4 and 8-fold for TEHTM, respectively, for Center 1 and Center 2. And for DEHT, the urinary metabolite concentrations were similar for the two periods. In parallel, we identified the exposure sources and the factors. We looked at the biometric parameters, which enabled us to identify the medical devices as the main source of exposure to plasticizers, particularly, as you can see on this, respiratory assistance, infusion therapy, enteral nutrition, and transfusion. Other factors were identified which would increase exposure to plasticizers, the most important being the premature state of babies, taking into account their age, their weight, but also according to the use of closed incubators. So to conclude, we can say that newborns' exposure to DEHP substitutes, particularly to TEHTM, leads to much lower amounts of the metabolites in newborns' urines compared to those of DEHP metabolites. Now, we also showed that DEHP is still very present in medical devices despite use restrictions such as overexposure, and according to the latest European ruling, particularly with regard to the suspected endocrine disrupting effect of or activities of DEHP. So the elevated internal levels of its metabolites in infants hospitalized in intensive care units, sometimes for several months, remain alarming. Additional efforts are necessary to promote a substitution of toxicologically critical substances such as DHP. That's still to come, particularly for respiratory assistance. Respiratory assistance leads to a lot of exposure and medical devices having a contact with blood, transfusion, ECMO, particularly when used in sensitive population groups such as newborns. in intensive care. So it's important to actually raise awareness and train health professionals, especially neonatologists. That's absolutely essential in order to manage this global risk linked to phthalates, and take into account the global environment, the global setting, and really state that the question is also, the question is, isn't it exposure to a panel of plasticizers is not leading to this cocktail effect that we have been talking about. Okay, thank you for listening to me, and I'm done.
Thanks a lot, Liz. Indeed, I'm afraid we are in the end of the session, but thanks already for having replied in the chat. Maybe I can just come back on a question, because I see indeed that your results, Liz, Lucas and Pauline, there are some interesting results that are overlapping, especially when it comes to the lack of data that we still need before assessing and stating that a plasticizers is a safe alternative. And maybe we can come back on this question. Lucas, maybe we can start with you about this missing data. Maybe we can sum up a little bit for the manufacturers, especially with attention to this European pharmacopoeia, because indeed the manufacturers are taking the European pharmacopoeia as a guidance, but this doesn't state that these are safe actually. And also, we are talking, for example, if we take the DINCH, I know the DINCH is already on the market, I think 20 years. So let's maybe summarize a little bit more what is missing from this pre-market study. And also, Lisa, feel free to jump in after,
Yeah, thanks for the other interesting presentations, and thanks, Lisa, for your overview. And I think the comment was from Tobias Johnson. Interesting comment, thank you. And indeed, as Katja said, that I think most producers base the possible use of alternative plasticizers on what is guided by the European Pharmacopoeia. But from, and I think most toxicology studies have been performed in vitro or in animal studies on exposure or ways of exposure that are not per definition the same as happens in clinical care. So I think the main missing data on that point from our perspective, from a clinical perspective, from end users, when speaking to nurses, to doctors and etc. in our hospital, or even from assessors of exposure in the lab, is that one, the legislative guidance, especially then basing on the medical devices regulation, there is, from that part, no guidance on any alternative plasticizer. Right? There is only for phthalates guidance to limit their use to 0.1 weight %, or if you say it's really necessary, to justify it and to label it, but there is no labeling of any alternative plasticizer. So if an end user wants to know, I want to use a product with this or that plasticide, there is no information. Only ways to use it in the lab and to assess what's the presence is there, which is for clinical care takes a few months to know. So academic, very interesting, but not in the end use. And then really on the risk assessment, I think the studies assessing the safety should be extrapolated back from exposure studies, as Lisa and Pauline and I showed right now, from relevant levels of exposure, relevant routes of exposure, and also for relevant toxicological endpoints. Most of the phthalates and alternative plasticizers have never been mentioned as neurotoxicants or pneumotoxicants, but several epidemiological studies show correlations between levels of those their metabolites in urine during pregnancy, or urine in early life, and asthma in later life, or cognitive deficit in later life. And our research group even showed that the level of phthalate metabolites in blood, in plasma, from children that are in the pediatric intensive care unit is correlated with cognitive deficit 4 years later. While it is never stated as a neurotoxic compound, big studies showed that there is a correlation for the children at higher levels during ICU care 4 years later. So I think there are plenty of gaps that need to be filled to be safe. Oh no, this one is safe. And I hope this answers your question or most of those questions on the topic.
So thanks a lot to all the participants and to the speaker especially. And yeah, I wish you a good afternoon. Bye-bye.
Okay.
Bye.